Okay, well, welcome everyone. Thank you for joining us today. I'm Tracy Smith, the Education and Outreach Coordinator for the Center for Integrated Cellular Analysis here in New York City. In this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. Today, we focus on new work using single-cell sequencing technologies to characterize antigen-specific CD8 positive T cell responses to COVID vaccination and disease. This work demonstrates how to measure and integrate diverse molecular modalities, including ATAC, RNA, protein, and more, all at single-cell resolution. We'll have time at the end of the talk for questions, and we encourage you to either raise your hand, and I'll ask you to unmute yourself, or you can type your question into the chat and I'll read it aloud to the speaker. So our guest today is Bingjie Zhang. Bingjie received her PhD from Tsinghua University in China, with her studies focused on epigenetic reprogramming during early embryo development. She's currently a postdoc in the CITE lab and is interested in applying single-cell technologies to better understand the underlying mechanisms of immune responses and their dysregulation in disease. And with that, welcome, Bingjie. I will hand it over to you.
Okay, thank you, Tracy, for the nice introduction. Let me share my screen. Okay, can you see it now?
Looks good.
Okay, I'm not sure. Okay, let's start. Today I would like to present our most recent work about how we characterize the antigen-specific CD8 T cells across the SARS-CoV-2 vaccination and infection using the multimodal single-cell technologies. The COVID pandemic has had a profound impact on human life, and thanks to the development of vaccines, they are being administered to help stop the global infection. And besides the human immunity mediated mainly by B-cell-derived antibodies, we should also pay more attention to the cellular immunity mediated by T cells, not only because the T cells protect after the infection and provide the long-term immunity memory, also because the T cells can recognize a wider range of the antigens, including these new variants. Upon exposure to the pathogen, some of the naive CD8 T cells would get activated and undergo clonal expansion while carrying its initial TCR sequences. These pathogen-specific T cells play a central role in the adaptive immunity, but generally speaking, they are very rare and difficult to isolate. Conventionally, there are several cellular assays we can use to assess the antigen-specific T cell responses. We can either incubate the T cells with antigen peptide pools and then measure the production of the cytokines, or we can directly scan the T cells with these dextramer or multimers. But both of these methods need well-designed peptides and the antibodies. And with the recent advancement in the single-cell technologies, one would expect to see that the single-cell studies could provide more insights into this topic. Well, the reality is, although there are a number of single-cell technology experiments like collected the PBMC before and after the vaccination, but none of them has clearly identified these rare antigen-specific populations, mainly because the T cells normally have the lower mRNA content, so it's quite difficult to clearly separate them out. So this represents us with a challenge topic, like how can we clearly identify these antigen-specific T cells and even track their future differentiation during the vaccination or infection. I want to say that this project is a close collaboration with the Dan Littman Lab in NYU Medical School, and also get substantial support from the NYU Vaccine Center. And great thanks to Ravi, a very talented postdoc in Dan Littman Lab. So let's take a look at the experiment overview. So basically, we took the PBMC collected from the 6 donors at 4 different time points. 1 day after the prime at day 0, 1 day after the prime vaccination at day 10, and also 1 week after the booster vaccination. We collected their PBMC and the CITE-seq and the ASAP-seq. So the CITE-seq can simultaneously profile the transcriptome and the cell surface protein, while the ASAP-seq can measure the chromatin accessibility and the cell surface protein in the same cell. Let's first take a look at the clustering results based on the CITE-seq experiment. As you can see here, with the assistance of the cell surface protein, we can cluster the cells based on the weighted combination of both their RNA and the cell surface protein. And we can also annotate their cell types at a relatively high resolution. And after we get these cells, if we split them into the different time points, we indeed can identify 2 distinct clusters, very small but distinct clusters that seem to be induced by the vaccination. They are barely visible at day 0 and weakly induced at day 10, but become much more evident at day 28, 1 week after the booster vaccination. And we can see the same phenomenon across the different donors. I want to emphasize that it can be quite challenging to bake up these cells if we only rely on the mRNA modality, as these cells get easily mixed up with other CD8 T cells. So, but with the addition of the cell surface protein, these cells can form a very distinct cell clusters. So after we identify these clusters, we want to ask, can we find any unique features about these vaccine-induced cells? And we found that for the group A and B cells, they mainly differed in the cell cycling genes as indicated here, but we also can identify a vaccine-induced gene module that's shared between the group A and B cells. Also, we can use about 197 genes as a gene module score and to identify the same cell populations in other data sets. And besides the transcriptional features, we can also find many unique protein expression in these vaccine-induced cells, including the CD38, HLDR, and ECOs. Those 3 markers are all very typical CD8 T cell activation markers. So with this transcriptome and cell surface protein features, next, of course, we want to explore their epigenome features. And to do that, at least we need to identify the same population in our ATAC-seq dataset. And we used a computational method developed by Yuhan Hao in our lab, the bridge integration method. So basically, we will use a multi-omic dataset which measures the RNA and ATAC-seq simultaneously in the same cell. So for each cell in the single cell RNA-seq data, we can use a weighted combination of the cells in the multi-omic dataset to represent it. So each cell in the multi-omic dataset is a dictionary element, and we can use a few cells to represent every cell in the single cell RNA-seq dataset. And also we can do the similar thing in the ATAC-seq dataset. We can identify a same dictionary elements to represent each cell in the single cell ATAC-seq dataset. And now we can easily, with this dictionary learning, we can easily translate every cell in the ATAC-seq dataset onto the space defined by the single cell RNA-seq dataset with this dictionary learning. And here is the result. As you can see here, we can use a TX multi-omic dataset. It's just the normal PBMC sample, don't need to be the vaccination sample PBMC. And we can use the ATAC-seq data from our ASAP-seq experiment and project onto the UMAP defined by our CITE-seq experiments. As I highlight here, this is all vaccine-induced cells we identified in the ATAC-seq dataset. We didn't see the population, the proliferating population in the ATAC-seq dataset, mainly because that's the chromatin accessibility difference is very subtle in the ATAC-seq in the ATAC-seq dataset. So we only identified the group A cells, these non-proliferating cell populations in this dataset. And once we identified this, once we find this cell population, next we also want to validate if this is true or not, because this is just a purely computational prediction. How can we know this workflow actually works? And the one thing I like the multi-omic dataset is that we always have an extra modality to help us validate the results independently. So as you can see here, all those predicted vaccine-induced cells indeed has higher expression with the CD38. It also has a higher gene model score, the vaccine-induced gene model score, which is calculated based on the chromatin accessibility. And consistent with our results in the CITE-seq experiment, these cells are barely visible in day 0, but get significantly induced at day 28. So these definitely are the same cell populations. And after we confirm the results, next we want to see if we can identify any enhancers unique to these vaccine-induced cells. And we can indeed find a strong induction of this enhancer upstream of the CD38 and echoes these typical markers of the vaccine-induced cells, like these two enhancers are unique to the vaccine-induced A cells. And what's more interesting is that we can also find about 13 SNPs, which are highly associated with the COVID-19 susceptibility, also reside in the enhancers unique to the vaccine-induced A cells, that I highlighted here and here. So it will be wonderful if we can prove in the future that the successful activation with these enhancers are important for the cellular immunity during the vaccination, during the infection, actually. So with these identified enhancers, we next want to see if there is any overrepresented motifs in these enhancers. And the BATF3 is our top candidate. And recently, there is also a paper reported that the BATF3 plays an important role in programming the CD8 T cell memories. And besides, this is a study in mouse, so maybe this result indicates that the BATF3 also play an important role in the human study. And besides the BATF3, we also nominated other few members from the IRF family and the TBX family that may also important for the development and maintenance of these vaccine-induced cells. So, and that's the results about the T cells. And I also want to share a very interesting findings when we explore the chromatin accessibility during in the myeloid cells. And we know that after the vaccination, our myeloid cells, like the CD14, CD16 monocytes, actually are the first group of cells that actually can be quickly activated. As I indicated here in the RNA dataset, we indeed can identify many interferon signaling genes that are quickly activated as early as day 2. But unlike the transcriptome, like we didn't see, it's kind of surprising for us to say that we didn't see a dramatic chromatin accessibility changes. As you can see here, these interferon genes get activated at day 2, as indicated here, but on the chromatin accessibility level, they are quite similar to each other, like barely the same. Like we also did a genome-wide examination to confirm this result. Like we plotted the scatter plot between the day 0 and day 2. You can see that they are highly correlated with each other. Like there is no, like we didn't expect to say they would be similar at this level actually. So maybe indicates that the landscape of the chromatin accessibility already are well established before the vaccination, so that the cells monocytes can respond very quickly to any external stimuli. So for now, we are sure these are the cells that get induced by the vaccination, but we actually haven't seen any strong, solid evidence to say that these cells are indeed the cells respond to the SARS-CoV-2 antigens. So how can we prove that? And to do that, we actually used a technology called the dextramer. As you can see here, this is a dextran, and we can load this with the SARS-CoV-2 peptide antigen here, and it's also carry a DNA barcode which is compatible with the 10x technologies. And what's more, it also has a PE fluorescent, so we can use it for the cell sorting. So we collected the PBMC at the day 28. Understand with the dextramer and also the cell surface protein, the same panel, and then and specifically enrich the cells for the dextramer positive or the CD38 and HLDR double positive cells. And we also spec in about 25% of the general CD8 T cell just to use it it as a control. And then we did the CITE-seq, which can simultaneously profile the TCR sequence and the cell surface protein, and also, of course, the transcriptional data. So here is a result of the CITE-seq clustering. The cells are clustered based on 3 modalities: the RNA, protein, and also the TCR. And with the help of the cell surface protein, we can clearly identify the same population, the CD38 HLDR double positive cells, and also the proliferating one and the non-proliferating one. And it's great to say that all those dextramer positive cells indeed largely overlap with the double positive cells, the CD38 and HLDR. But if you take a close look at this overlap, we can find a subset of the cells that carry the high signal of the CD38 and HLDR, but devoid of the dextramer positive signal. So these are the populations that are different, and we kindly group them based on the expression of the KLRG1. Like we think that they are actually the 2 different cell types, and based on the following evidence. So we called this cells with a high expression of the KLRG1 as a bystander cells, and these are the antigen cells, and we call them based on the following reasons. And besides the high expression of the KLRG1 in bystander cells, and we also, if we break down into individual donors, and it is true that the bystander cells generally have a much lower fraction of the dextramer positive cells. And to exclude the possibility that these cells probably just harbor the TCRs can't recognized by our dextramer panel, we also downloaded TCR beta sequences in these 2 public large dataset that's unique to the SARS-CoV-2 peptide antigen. And consistent with our dextramer results, you can see that still these bystander cells carries a much lower fraction of the cells matching the public dataset. And also, the clonal expansion ability is a key feature for these functionally antigen-specific cells. And you can see that compared with the antigen cells, these bystander cells indeed have a much lower fraction of the cells in hyper and large clonal expansion cells. So we also confirmed this, like the identification of the KLRG1 cells in the flow cytometry. And you can see that consistent with our CITE-seq experiment, like in the KLRG1 negative cells, it's also very rare in day 0, but significantly induced after 1 week after the booster shot at day 28, like they are significantly induced. So it's great to see that we can also confirm the results using the flow cytometry. So for now, all the data I showed you before is the data that we generated. And as I said before, if we can clearly identify the unique features of these vaccine-induced cells, in theory, these cells can also be identified in other data sets. So we took, and to validate this, we took a data set from this study. And this study haven't identified these populations in their results, but if we map their dataset onto our CITE-seq reference, you can see that the clear emergence of this rare population, as I indicated here, and consistent with our results, is also in their dataset is also gets significantly induced 1 week after the booster shot, and has a consistent expression of the CD38 and the ICOS in these cell types. So that's great. We indeed can use these unique features to identify the same populations in any other external data sets. And it's quite interesting to think that if these cells are the antigen-specific cells, besides a vaccination, in theory, we should be able to identify the same populations in the real infected samples. And to do that, we took a data set in the SARS-CoV-2 acute stage, and they also use the same dextramer as we used in our data set to accurately label these antigen-specific cells unique to the SARS-CoV-2 antigens. So you can see that in their data sets, they can't form a distinct cluster. Also, they have the dextramer information. But it's great to say that now we know the ground truth. it's great to see that even with the gene module or without any protein information, with this gene module we identified in a vaccination dataset, we can accurately predict if the cells are dextramer positive or not, to say if these cells are really antigen-specific or no. And as I showed here in the heat map, you can see that these dextramer positive cells indeed express the same vaccine-induced gene module. So it's great to see that these vaccine-induced cells share the same transcriptome in the vaccination and also in the disease. And these results give us much more confidence that we indeed can use these unique gene features to identify the same populations in vaccination or in the real infection without the further assistance from the cell surface protein. And we know that these antigen-specific cells play a very important role in the adaptive immunity. So we can recur to say if these cell populations correlate with the disease severity. And to test this hypothesis, we took a data set from the combat. It is a large data set collected from about 100 donors, like they are infected with the COVID-19 at the different severity. And we only take the CD8 T cells and did the clustering. And with the cell surface protein information, we can annotate the cells as the same cell types as we did in the vaccination, the antigen proliferating cells, antigen cells, and also we identified the same populations as KRD1 positive cells. And once we split the data set based on the disease severity, it is clear that in the disease samples, these antigen-specific cells increase significantly, compared with these healthy controls. And one particularly interesting findings is that we found the abundance of these antigen cells is highly predictive of the patient's future outcome. Like if we took all the donors and the severe disease and group them based on whether on their future outcome, whether they recover or get worse. Like you can say that actually the two groups are quite different in terms of the percentage of these antigen-specific cells. Like in the deteriorated groups, they tend to have a fewer percentage of a fewer fraction of these antigen-specific cells, indicating that these cells are indeed very important. Mounting these CD8 T cell responses is indeed very important for fighting against the infection. Well, in the context of the infection, I think one intriguing question is that can we can we track the future differentiation of these cells, like what they differentiate into in the future? And even with the recent advancement, it's actually quite difficult to do the lineage tracing or cell labeling in the in vivo samples, especially for human. But now, if we can know what cells are the antigen-specific ones, and combined with their TCR information, this inborn cellular barcode, we can actually have a very simple but powerful way to pick these cells out. Like we can simply do the overlap between using their TCR information, as I shown here, and we actually found it quite surprising to see that the differentiation trajectory is quite different in the mild and the severe disease. As I shown here, like in the mild samples, there are about 40% of the TCRs in the antigen-specific cells can be identified, can also be found in the cytotoxic cells. But in the severe group, this percentage decreased to about only 20%. And this result is also true if we split the result based on the split into individual donors. And you can see that in the mild disease, the overlapped TCR percentage is about 20 to 25% in the mild group, but decreased to only about 10% in the severe and critical groups. And we also picked up all the cells that carry the TCR identified in the antigen-specific cells and plotted their distribution in the UMAP. And you can see that unlike the mild group, in the severe group, all these TCR, all the cells carry the antigen-specific T cells tend to differentiate more into the timer cells, unlike the myeloid cells, like they are more like tend to differentiate into the cytotoxic cells. So this differentiation of the antigen-specific cells is skewed toward the timer cells in the severe disease. The timer cell is more relevant to the cell type of the aging. So maybe can explain why these cells tend to be more severe in the severe group. Yeah. And we also confirmed that this result is not driven by the disease severity and the symptom, the duration of the symptom. You can see that they are basically, you can't see a clear correlation. So the differentiation difference not driven by like the severe groups may have longer symptoms of COVID infection. It's not. At least, yeah, the showed here, we think it's not. Yeah, so in summary, in this project, we clearly identified the antigen-specific T cells in the vaccination dataset, and we also nominate potential enhancers and transcriptional regulators. And more importantly, we found that the CD38 and the HLDR double positive cells are actually heterogeneous, and in the future, the inclusion of the KRG1 cell surface protein is not a bad idea when we do this flow cytometry to more clearly identify the cells that are capable of the rapid clonal expansion. And also that's the real antigen-specific cells. And what's more, we also identified the same population in the real infected sample that shows that the frequency and differentiation outcomes are predictive of the subsequent clinical outcomes. So that's the main content of my talk today, and great thanks to everyone in Scientific Lab and the LISMA Lab, especially special thanks to Rahul and Yuhan and John in our lab. Yeah, thank you. Let me know if you have any questions.
Great. Thank you so much, Bingjie. What a great talk. So yes, well, let's move on to Q&A. If you would like to ask a question, please raise your hand or type your question in the chat box. And I see Bruce, feel free to ask your question.
Oh, hi, thank you. Really interesting. What do you think about the aging attenuated vaccination response? And so if you look at that, if you can get your hands around the aging-based attenuation of vaccination response, is that the same as what is the severe component, or is this really a separate differentiation type? And the other thing I was thinking about is what's happening on repressor T cell side. Do you see any kind of a differential loss of repressor T cells in the more severe case where you're getting more cytotoxics coming out?
When you say aging, you mean the age of the donors?
No, if you look at it like in the aging databases, you'll see that there's an attenuation of the magnitude of the T cell response that is with age, so it goes down with age. Is that the same as this hyper CD8 T cell? I'm wondering, it could well be different. And then the second is like on regulatory T cell effects that are going on here. Do you see like more of a loss of Tregs in this more severe response, CD8 response?
Yeah, that's a very interesting point. Actually, I think they are probably similar in the same way. Like this is just the T cell response, no matter in the aging samples or the infected samples. The field induction, like the field activation of this CD8 naive cells, may indicate somehow the impairment of the T cell response, right? So I think it's reasonable to say more terminal cells in the aging samples or the more terminal cells in the severe disease. Yeah, probably the same. Yeah, okay. Further to take a look at the aging samples is quite interesting.
Thank you, everyone. Thank you, Bingjie, so much. It's been a great talk. And thank you all for joining. I'd like to remind you the seminar has been recorded and it will be available soon on YouTube and on our website, multimodalintegration.org. You can also follow us on Twitter at segs_ICA or check our website for the latest announcements regarding upcoming talks and events. With that, I'd like to thank you all for joining, and we hope to see you at our next seminar. Have a lovely day.